in this lecture video, I'm going to talk about fermentation. Fermentation will occur if an organism, for some reason, can't carry out that cellular respiration process that I talked about in the last video. So if there's not a final electron acceptor, or if the organism doesn't have the genes, they don't have the DNA to make those enzymes or carrier molecules that they need in the electron transport chain or in the Krebs cycle, then they may switch over to this fermentation pathway. So in the fermentation pathway, the NADH molecule, which is one of our carrier molecules, has to be reoxidized back to NAD+. So we're gonna have to remove that electron so that it can serve again as an electron carrier for glycolysis in order for it to continue. So the organism is going to go through glycolysis, but then for whatever reason they can't go into those other pathways, the Krebs cycle or the electron transport chain. They've got to find a way to remove the electron that was taken in glycolysis so that that electron carrier can be reused. And so some organisms can use pyruvate or another organic molecule as a final electron acceptor in the process of fermentation. Okay, so fermentation does not involve the electron transport system. Therefore, it's not going to produce any additional ATP. Remember, the electron transport chain is where we get the majority of the ATP that is produced. So if an organism goes through aerobic respiration, where oxygen is serving as that final electron acceptor, we can get 30 to 34 ATP from the electron transport chain and add in the ones we got from glycolysis in the Krebs cycle to get about 38 ATP. So we get a lot of usable ATP. In anaerobic respiration, when oxygen is not the final electron acceptor, when we're using some other type of inorganic molecule like nitrogen or iron, we're still going to get the 2 ATP from glycolysis and the 2 from the Krebs cycle, but the number that are generated, the number of ATP that are generated in the electron transport chain varies. So we can end up with 5 to 36 ATP. So anaerobic respiration is not as efficient as aerobic respiration. Then when we look at fermentation, we're getting those 2 from glycolysis, and that's it. So it's the least efficient of all of the methods of metabolism. However, fermentation is beneficial for us. We use fermentation to produce a lot of different types of foods and other products, including pharmaceuticals. We also can look at the products of fermentation to identify bacterial species or other microbes. So in some species, they produce lactic acid. So our lactobacillus, which is the primary bacterium that we find in yogurt, produces lactic acid as a result of fermentation. And so this is a beneficial process because we eat those products. We eat the yogurt, we eat the sour cream. But lactic acid fermentation also occurs in animals, in muscle cells. In the last video, I mentioned that, if oxygen is not available or if we run out of oxygen, then we switch to anaerobic respiration. But we can also switch over to this fermentation pathway. And in the fermentation pathway, we produce lactic acid as a waste product, and that's what makes our muscles sore after we've done some, extensive exercise. So when lactic acid is the only fermentation product, we call that homolactic fermentation. But a lot of bacteria perform what is called heterolactic fermentation, where they can produce lactic acid or acetic acid or ethanol, carbon dioxide. They can produce several different end products. Alcohol fermentation is another form of fermentation. This produces ethanol, and this reaction is shown here, where the pyruvic acid is converted to carbon dioxide and acetaldehyde, and acetaldehyde is then converted into ethanol. And keep in mind, we're not showing the enzymes that make these processes occur, but those are there. Remember, enzymes are necessary for these processes to occur. We also get the stripping of that electron to convert the NADH back to NAD+, so that it can be used in glycolysis again. So ethanol fermentation, of course, very important in the production of alcoholic beverages, as well as bread. When we make bread, the CO2 is what makes the bread rise, and then the ethanol is, burned off during the cooking process. For alcohol production, the carbon dioxide is what makes it fizzy, okay, what makes it, carbonated. And then, of course, we get varying ethanol contents depending on what organisms are used and how long the process occurs. There are other fermentation methods, but again, the goal is to convert our NADH back to NAD+ by removing that electron. Without this process, we wouldn't have this NAD+ electron carrier to go back to glycolysis and pick up that electron. So the fermentation pathway, important in those waste products because we can manipulate that. And the bacteria may use that for other purposes. But the most important thing is just to get back to NAD+. So here are some other fermentation pathways. We can get butyric acid that we can use to make butter. We can get different forms of acids, acetic acid, for instance. We can use that in vinegar. That's what vinegar is. It's just acetic acid that's been put in water and reduced to a 5% concentration. Propionic acid we use to make Swiss cheese. That's what causes those holes in Swiss cheese. So we have a lot of different types of fermentation pathways that the microbes can use. And again, these we can manipulate so that they benefit us.